So in these series of videos, what we're going to be doing is going through the January 2026 New York State Life Science Biology Regents Exam. In the first cluster, I'm going to talk a little bit about some test taking strategies, but after that, I'm going to focus really on the rating guide and how to score yourself once you've done this cluster. So that's the plan. This is what the Regents exam will look like. You have the instructions, some information about calculators, communication devices, and then you get right into this first cluster. So the way that these exams are set up, it's a series of clusters of questions that all sort of are focused around one theme. And so what we see when we look at this first cluster was this is the first 5 questions. They're all based on this next set of information. So my guidance is always to be really careful in reading everything that you see in the cluster. And so I'm going to just do that with you a little bit. So the title of this is Two Kinds of Twins. I'll read over this introduction. Twins are 2 offspring produced by the same pregnancy. The most common type of twins are identical twins and fraternal twins. The genes of fraternal twins may vary as much as non-twin siblings, whereas identical twins share the same DNA. However, as identical twins age, their physical characteristics can show differences that distinguish them from each other. Okay, so we have a little bit of information there. If you like to use highlighters, it might be helpful to highlight key information that you see there. But what is so important is you have to read these things. A lot of times students will just kind of skip over or skim the introduction to these clusters or not really read it, but there is often key information that you need in these introductions. So we've read that. We continue on. The model below represents some information about twin development. So if we look at the model, you always want to really analyze the model, see the information they're giving you, try to really understand what this model is showing before you even get to the questions. So here, when I analyze this model, we have 2 sides, model A here on the left, model B on the right. Okay, so I see here 1 egg and 1 sperm, and I have a graphical depiction of those. On the other, model B, I see 2 eggs and 2 sperm are labeled, so I can also see them visually. There's 2 different egg cells, 2 different sperm cells. Then I continue analyzing this model. I have an arrow, which usually represents something happening, a process happening. These 2 arrows are both labeled X, okay? Then I have a picture. It's showing me the womb. It's showing me the uterus. It's not labeled, but that's what this is. Okay, and so in model A, I see that there's 2 fetus there, and they share the same placenta, but they have separate amniotic sacs. Okay, if I look over here at model B, what I see also separate amniotic sacs, but also 2 separate placentas. So that's something different between model A and model B. Model A, in which you have the same placenta, model B, you have 2 separate placentas. Then as we continue on, we have sort of a magnification. This is showing the leg of a fetus, and it's showing muscle in the leg. Okay, so I've analyzed that model. I understand what it's showing me, and now I'll start to get to the actual questions. So question 1, use the information in the models to identify which model, A or B, represents fetuses that would continue to develop into identical twins, and identify 1 or more structures in the model that you selected that could be used to support your answer. Okay, so I have to choose model A or B first, and there's a place for me to write that in here. So use information in the models to identify which model, A or B, represents fetuses that would develop into identical twins. Okay, so when I look at there, I just have to pick model A or B. The correct answer is model A. Identical twins come from a single egg and a single sperm cell. And so that's what I would choose. And that's actually supporting my answer as well. So I could say model A. And what I might say, if we look here at the scoring guide on the other side, let's look at what they say are appropriate responses. Allow 1 credit for model A and correctly identifying a structure. So we can say it's an identical twin in model A because there's a single fertilized egg, or that there's only 1 sperm cell and 1 egg cell creating those fetuses. We could also say, looking at this part of the model, they share the same placenta. That's evidence that they would be identical twins. So any of those answers would be considered correct. So if you're grading yourself and you got model A and you gave appropriate evidence, give yourself +1 here in the margin. That's the first question of this cluster. Then we get to the second question of the cluster. Which statement describes how a process that occurs at X, okay, let's go back and look. Okay, so it's X is this arrow going from a egg and sperm cell, a fertilized egg, into these twin fetuses. The same thing is true here. We have 2 eggs, 2 sperm, 2 fertilized eggs, also going through process X to produce the 2 twins. Okay, so which statement describes how process that occurs at X within the models results in specialized cells in the fetus's leg? Okay, so you think back, okay, and you try to think about, okay, what is happening at X? How does a fertilized egg grow into an actual fetus? And hopefully you're thinking, well, that's cell division, that's mitosis. Mitosis is what takes a single fertilized egg cell and allows it to grow through many stages into one of the fetuses here shown. Okay, so that's what's going on there. And when we look at the answers and we read each of them over, okay, I always encourage students to read the question, then try to think of what an answer might be before you actually read the 4 choices. So when you read those 4 choices, you should find the correct answers. 3, a cell in a developing fetus can divide into muscle and bone cells with the same DNA. But we know that because of differentiation, some of those genes are turned on or off when they're found in the leg. And so that's why these cells that are developing in the leg have different shapes, different functions, even though they have the same DNA as every other cell in the fetus. That's how our genetic code works. Every cell has the exact same DNA, but depending on the location and the function, certain genes are turned on and off, allowing them to differentiate into different types of cells and tissues and organs. So the answer to question number 2 is 3. If you got it correct, put a +1 here. We're going to be keeping track and adding up all of our points. Okay, then we move on to question 3. Use evidence and reasoning to defend the claim that processes that occur during sexual reproduction result in fraternal twins having different traits. So now we're focused on those fraternal twins and why they have different traits. Okay, and we're thinking about sexual reproduction. And so if we look here at the scoring guide, we have some correct answers. Okay, this first one, fraternal twins result from 2 eggs and 2 sperm cells, which carry many different combinations of chromosomes. Due to the mixing of genes during meiosis, the twins will inherit different traits from each parent. So that's a complex answer. It's a really great answer if someone were able to provide that. A shorter, simpler answer that's also correct, fraternal twins that have genes from different sperm and egg cells will have different traits because the genes come together in different combinations when sex cells are produced. So you don't have to have those 2 answers word for word, but in order to get it correct, you should have something that's pretty close to that something that refers to 2 different sperm cells, 2 different egg cells create fraternal twins, and those sperm and egg cells are each unique due to this process of meiosis. Every egg cell and sperm cell created through the process of meiosis has different sets of chromosomes, different genes, and that's where the variation in sexual reproduction comes from. So read your answer, see if you would have earned credit on number 3 based on those guidelines. And we're going to continue down. Okay, so now what we have in this cluster is some more information, more models, some more reading passages. So you always want to focus, you always need to read every piece of information given, and you always need to analyze every model. So I'll do this with you for this first cluster. Scientists are investigating how the expression of genes in identical twins may be altered by epigenetic changes. Now you may see this and you may say, epigenetic changes? I've never heard of that. Well, it is covered in the lactase lab a little bit, and we'll see that in a minute. But if you see a phrase that's unfamiliar, don't get concerned, because what you'll see here, they use the term epigenetic changes, but then in the next sentence, they're actually defining that term for you. Epigenetic changes occur when certain chemical groups, such as methyl groups, attach to a cell's DNA and influence the expression of particular genes. So it's defined it for us. So you don't actually need to have that epigenetic term memorized because it's being defined in this introduction. Environmental factors and lifestyle choices, such as diet, have been linked to epigenetic changes. The model below shows how methyl groups can attach to DNA. Okay, so we see in the model, I want to analyze it. I see this is DNA. I can tell by the shape, it's also labeled. And this is methylated DNA. And what I see is there are these shapes have been added to the DNA. Those are methyl groups. Okay, so that shows me methylation. Okay, we continue then. Electrophoresis, that's a process you likely learned about in class, but it's defining it for you here as well. Electrophoresis is a process that can be used to compare DNA segment migration through a gel. And we see these are some examples of electrophoresis. The image below shows a comparison of DNA taken from body cells of 3-year-old identical twins and 50-year-old identical twins. Okay, so let me look at what the model is actually showing. What is electrophoresis showing? So we have the two twins, twin A and twin B, okay, those identical twins. They were the 3-year-old twins. I see that labeled at the bottom. When I look at the electrophoresis, it pretty much to me looks exactly the same. Then I look at twin C and D, the 50-year-old twins, and I look at their electrophoresis pattern, and I definitely see differences between twin C and D. Right, twin D has these bands here, and twin C doesn't, and same thing here. So there are definitely differences that I'm seeing in the electrophoresis pattern in these older twins versus the younger twins. So let's continue on and read the questions. Number 4, a researcher wants to determine the cause of the amount of variation seen between identical twins at 50 years old when compared to the twins at 3 years old. So I'm thinking, okay, 50-year-olds had differences in the electrophoresis pattern. The 3-year-olds didn't really have differences. So which question should they ask to help them determine the cause? So when I read this, it's asking me for a question. So if I read over these questions, what I'm going to see is the first answer: how does the number of methyl groups attached to the DNA of 50-year-olds compare to 3-year-olds? That would be a good question to help us understand what's going on here. Are these differences due to methyl groups? That would be a great question that would help us understand. We could see over here in the rating guide, number 1 is the correct answer. So give yourself 1 point if you got it correct. And then question number 5, the last question of this cluster. One of the 50-year-old twins, twin C, has been identified as being at risk for developing diabetes because of a specific gene has been methylated. The same gene in twin D does not show signs of this change. How might genetic technology be used to prevent twin C from developing diabetes later in life? Okay, so when I read over the answers, using splicing enzymes to remove methyl groups, developing medications that could remove methyl groups to allow normal gene expression, developing medications that would add methyl groups in this region, or using these splicing enzymes to add methyl groups. Okay, so when I read the question's asking how I could use technology to prevent twin C from developing diabetes. So the only one of these, so splicing enzymes would be cutting the DNA. That wouldn't be helpful. And so when we're looking at choice 2 and 3, what we're seeing is choice 2, if we think these methyl groups might be interfering with gene expression, then maybe we could remove those with a medicine. And so 2, if we look over here, is the correct answer. And so number 5 is 2. Give yourself a point in the margin if you got that correct. So you've successfully completed the first cluster in this practice exam. You've graded yourself. And what's going to happen in a next set of videos, we're going to continue to go cluster by cluster. I'll speed things along. I'm not going to read through each question with you and really focus on the scoring so you can get a good idea about if this were your Regents exam that you were taking in June, how would you have done? How would your score have scaled? And would you be meeting the goals that you have for this course? Thanks.